Hello, welcome to the final episode of this first session of the EVOS seminar series. Our guest today is Dr. William Ratcliff, who's going to be presenting his research on evolving multicellular snowflake yeast. And this is one of the most innovative experiments I've seen. It's got really big implications. So take it away.
Cool. All right. Thank you, Max. It's a pleasure to be here. Can you guys hear me okay?
Yep.
All right, great. So let's see. Sorry guys, I need to go full screen on this guy, and then I'm not screen sharing yet, right?
You are not screen sharing yet.
All right. Screen share. Share and full screen. All right. Can you guys see my talk? All right, great. So I'm actually really glad to be speaking to a class of people studying major transitions, especially here at the end. And I would love at the end of this lecture to kind of try to connect back to other lectures that you had, discuss some of the sort of fundamental philosophical and biological issues that have to do with the major transition. Are we really seeing one in the lab? But as you guys know, major transitions are both important and hard to study. I'm abstracting the normal introduction that I give here, since you guys already know about this topic. And the basic reason that they're hard to study is because they're relatively rare and they're relatively old. Multicellularity is a great example of that. Actually, it's a great example of how it's not the rarest thing in the world. There's at least 25 different independent transitions to multicellularity, but even the sort of new ones, quote unquote, you can't see me air quoting over here, are relatively young. Relatively old. The new ones happened a long time ago. So, Volvocene algae and brown algae, so the kelps that you see at the shore, both evolved sometime in the Triassic, so 200 million years ago. And so that's a pretty long time ago, and we don't really have necessarily the ancestors of those things around to study. We usually don't have early transitional forms available to us to study. We have things that are relatives of a lineage which became multicellular that are still single-celled, and things which are now very complex. And so understanding those first steps is critical. That's where a lot of the sort of evolutionary conflict and stuff, if it's going to arise, can arise. Understanding how those conflicts are resolved and how things evolve to be more complex is an important thing, and that's what we're studying in the lab. So to give you some ideas of basically the questions which have motivated our work. And I also want to say, if you guys have a question, feel free to jump in. It's kind of weird. I can't really see you because of the way that PowerPoint is showing me two versions of my slide.
We can interrupt if need be.
Great, yeah, please do. All right, so question one, can simple multicellularity evolve rapidly? And I think, the fact that I'm even here, the title of my talk, tip my hand, so I'll show you a little video. This is a snowflake yeast. It's a multi-celled fungal critter that we evolved in the lab from a single-celled yeast. And here it is under time-lapse microscopy, reproducing, popping out these little baby propagule snowflake yeast. They have this simple life cycle where these snowflake yeast basically grow back. Here, I'll play it again. They split off from the parent, and then they begin growing. And when they get to about the parent size, they begin splitting themselves. So they have a very simple emergent life cycle. All right, wrong direction. Oh, and here's just a cool picture of them in 3D from my collaborator's confocal microscope. And you can see they have this kind of cool branching morphology. Turns out that it follows really nice, elegant mathematical rules. I'll talk more about that later. So second, how do these multicelled clusters form? And does the way in which they form matter? Third, how do they gain the ability to evolve as multicelled individuals? This is kind of the fundamental evolutionary question, right? If single-celled things have spent, 1 billion or 2 billion years adapting as a cell, well, how do they now form a group and lose that former level of cellular individuality, evolving from a former organism that was the recipient of many adaptations into a part? How are they sort of stripped of their evolutionary autonomy? And finally, how do they evolve to be more complex? I think we can provide some answers to all of these questions. So like I said before, multicellularity has evolved a number of times in really diverse ways. These are all examples of independently evolved multicellular lineages. So we have animals and plants, the ones that everybody thinks about. Some people tend to think about slime molds. People in, I'm sure, your class are people that would have thought about slime molds. There are multiple transitions within the fungi. Bacteria are actually the first things that have gone multicellular. This is actually a Myxococcus colony, fruiting body, I guess I should say. We have brown algae, red algae. Red algae were, now it's up for debate, they're either the first or the second multicellular eukaryote. We have good fossil evidence that they evolved around 1.2 billion years ago, but now there's new evidence that filamentous fungi beat them to the punch by about 400 million years. And then we have, of course, multiple transitions in the green algae. Now, one of the issues with multicellularity is that when I say the word multicellular, you have something which pops up in your mind. And those things tend to be large, complex, charismatic. You tend to think of plants and animals. I don't blame you. That's what everyone sees. And one of the issues here is that there's really no sort of generally accepted definition of what it is to be multicellular, or even what defines multicellularity. Although I think, and so people tend to kind of use criteria which fit the model system that they're working with. This is not something which is unique to major transitions research. This is just something which tends to happen with scientists that study model systems. But I think everybody would agree that multicellular organisms share a set of traits. They all have multiple cells, and almost always those cells are physically attached. They have some sort of multicellular life history. And by that I mean they reproduce, and those offspring, because they're much smaller than the adult, are usually or always juvenile, so they have to grow before they become reproductive again. And critically, adaptation takes place in the context of this multicellular life history. Or to put it another way, single cells are no longer the focal point of adaptation, groups are. And so, selection is acting among groups, and groups are gaining group level adaptations in response to that selection. I mean, that's really important. Otherwise, from an evolutionary perspective, it's hard to make an argument that they're a multicellular organism. And this is the only one where I think there's potential disagreement, but most people would argue that multicellular organisms are characterized by some degree of cellular specialization. But I think you can also make an argument that cellular specialization is the result of the evolutionary process I was just describing it here, and therefore is an outcome of multicellularity, not a prerequisite. So there's no question that life on Earth has become more complex over geological time. And key steps in the evolution of this complexity are termed major transitions in evolution. I'm going to skip over this because you're in a class on this topic. And one of the things that's kind of interesting is that the sort of fundamental theory of evolution doesn't predict increased complexity over time, right? It makes concrete short-term predictions of increases in average population fitness, but that doesn't necessarily translate to increased complexity. And so, it's also sort of worth noting that there's really no evolutionary prerogative in a Lamarckian sense to become more complex over long periods of time. Many lineages have stayed just as complex as they were long ago. So cyanobacteria, for example, still form stromatolites, much like they did 3.5 billion years ago. Crinoids have changed little since the Silurian, and horsetails have changed little since the Devonian. So how did we get from a world dominated by microbial life of many, stripes and shapes to one which supports such a breadth and abundance of complex and large and charismatic multicellular organisms? Well, we don't really know, and that's partly because multicellularity is a challenging problem to work on. Like I said sort of right at the beginning, known transitions to multicellularity are old. These are examples of brown algae and Volvocene algae, which are both sort of the new kids on the multicellular block, evolving multicellularity around 200 million years ago or more. These things don't tend to fossilize very well. Soft, small, microscopic, soft-bodied creatures are not what you find when you go to a fossil bed. You find shells, or bones. For a reason, those are tough, recalcitrant structures. But that doesn't mean that we don't have fossils of things which could be multicellular. In fact, we have these beautiful fossils from the Deshantu fossil beds, many of these microfossils in which you can clearly see cells, and you can kind of even get an idea of how they're dividing and what they're doing. But even when we have these rare glimpses into the biology of, of sort of possibly, just pre-Ediacaran times, right? Like right before the Ediacaran and the Cambrian explosion, you find these cool microfossils, and yet we don't really know what they are. There's been a lot of disagreement about what these microfossils represent. Are they animal embryos? Are they Volvocene algae? This is an actual hypothesis that they're Volvocene algae. That's probably not true, but the fact of the matter is we don't really know what they are because we can't really probe them other than looking at their structure, and that doesn't tell us all that much information. So one of the things that the community has done for great reasons, it's actually been very informative, but it's also been sort of their hand has been forced, is basically looking at lineages where you can take a multicellular group like animals and take a very closely related unicellular lineage, which never became multicellular, like quinoflagellates, and comparing them together and saying what unicellular and the multicellular lineages that share a recent common ancestor, although recent is, I'm again doing air quotes because, quinoflagellates split from the lineage that made animals probably more than 600 million years ago, so that's a very long time. And the problem here is that while you can find many differences between unicellular and multicellular representatives of a single group, it's hard to know what to make of that because the temptation is to assume that those single-cell taxa are similar to the ancestor, or at least much more similar than the multicellular one. And then, say like, well, the differences in the multicellular one may reflect what made them multicellular. But they each had a very, very long road in which they've been changing, and we can't assume that those unicellular taxa really are representatives of the ancestor. They've had a long time to adapt to single-cell taxa, and in fact, they're probably evolving. I mean, we know they're evolving. they're probably changing substantially. And that makes it very difficult to disentangle cause and consequence. So for example, is the much larger genome of animals compared to quinoflagellates, is that increased genome size a reason that they went multicellular in the first place? Or is it a consequence of 600 million years of multicellular adaptation? It's hard to pull these things apart. So to explore, the origin of multicellularity and really just the first early steps, we have two choices. We can either get in a time machine, go back 600, 700 million years and observe this process as it's happening. To be honest, if I had a time machine, I would probably go back and look at great white sharks the size of buses and dinosaurs and all sorts of really cool multicellular critters rather than tiny clumps of cells. But it's a moot point because time machines don't exist. So instead, we're trying to do time travel in the laboratory.
Can I ask a historical question?
Yeah.
So now there's feedback. Sorry, I have to. So you pointed out like 6 or 7 multicellular lineages. Is it just a product of the way it's taught, or did the 3 major kingdoms, animals, plants, fungi, did they all achieve multicellularity at a somewhat similar time? Like, is there something about 700 million years ago that was important, like in terms of like the rest of life, or is that just an error in the way that I've learned the chronology?
So okay, that's a fantastic question. So it turns out that the origin of multicellularity in those lineages is kind of the opposite of what you might expect based on just how complex they are. So it turns out that the youngest of those 3 is animals. Sorry, is plants. The youngest of those 3 is plants. The oldest is fungi, and animals are in the middle. So there's a recent paper showing that filamentous fungi basically date to right around the origin of eukaryotes. So like 1.6 billion years. It's a really long time. Now that's filamentous fungi. That's not mushroom-forming fungi. But still, that's a simple form of fungal multicellularity. Turns out animals, we don't really have a great estimate for when multicellularity first arose, but it's somewhere in the time frame of probably 800 to 700 million years ago. And then plants, land plants really kind of arose in the mid 400s of millions of years ago. And let's see. So turns out that the vast majority of eukaryotic multicellularity does date to the last billion years, which is interesting because they had a lot of time before that, right? At least 600 million years where there's eukaryotes and a big planet and tons of different environments, and you'd think that there's advantages associated with size and that they should begin evolving multicellularity much earlier. We know of bacterial multicellularity, so cyanobacteria with simple but honest to goodness division of labor, nitrogen fixing division of labor, spore forming division of labor. They were doing it like 2.5 billion years ago, so a really long time ago. But really, things get interesting and really kick off in the last billion years, and I'd say the unproven or somewhat poorly substantiated but plausible hypothesis for that is that it's the rise of atmospheric oxygen. So we have oxygen going from roughly 1% of modern day to close to modern day, starting at 1 billion years ago and taking a few hundred million years to crank up. And I think that that's certainly a major limitation. High concentrations of oxygen are a major limitation for the kind of multicellular life that we exhibit. If you're going to be a large aerobic thing with a big body mass, you need a circulatory system and you need a lot of oxygen if you're going to be a respiratory critter. But that doesn't apply as important. It's not as important if you're small, because you have a high surface area to volume ratio. That's not as important if you're, say, photosynthetic. And so I think that's really not important at all if you're fermentative. Although you do need a high carbon flux to be a big fermentative thing. So I think that applies to some things like animals, but I don't think it's the whole story. And nobody knows the whole story. Nobody knows really why in the last billion years you see pretty much the majority of eukaryotic multicellularity taking off. And part of it is, and in fact, it may be a confluence of factors. I'm just speculating here, but my guess is that part of it is that it does allow things to have big bodies with minimal cost. And you probably get a lot more ecological complexity driving essentially positive feedback loops. That if something is evolving to be multicellular or more complex, you get arms race dynamics, you get predator-prey dynamics, you get all sorts of things which drive similar increases in complexity in other lineages. And so what may be a trigger for, say, animals may also have ripple effects through other lineages. But I don't think it's the whole story. So some of it might just be time. I don't know. Nobody knows, as far as I know.
A quick follow-up question. Those filamentous fungi, they must have been aquatic, oceanic, right?
Yeah, I think so.
So are there any examples alive today of multicellular fungal forms that live in the ocean?
Well, yeah, I'd have to actually double check that paper to see where they found the fossils. I wasn't paying attention to environment as much as just looking at the traits and being like, are they sure these are archaea? Because
Yeah.
Yeah. I don't know.
All right.
I don't know that much about marine fungi, but I'd be shocked if there were not marine fungi. There's so much stuff decaying in the ocean.
Yeah.
Fungi have
They're 444.
444
extant species of marine fungi.
Ah.
It looks like. I don't know.
Thanks, Wikipedia.
I'm just on Wikipedia, but we'll see.
Nice. Nice. Cool.
Had to do it.
Yeah, no, that's good. That's good. Any more questions, or should I jump back into the spiel?
Let's keep going.
Cool. All right, screen share. How's the frame rate on my talk?
It's not great, and you're exceptionally blurry, but the audio is good, so
Dang.
We'll make
bummer. Some of these videos are really clean. You see them in like 30 or 60 FPS.
Yeah. One day in the future, this is gonna look real silly, but
yeah, that's cool. So anyway, our model system, our main model system is yeast, and that was chosen for pragmatic purposes. It's a single-celled eukaryote. It's facultatively sexual, so you can turn on and off sex, which is great for genetics. It's a small and very well understood genome. It's highly mapped and there's a lot of functional annotation, and it's very easily grown and very easily genetically engineered. All right, so the first step in evolving to be multicellular is evolving to form a group of cells. Now, in nature, this might occur if you, say, have a predator which can consume single cells but has a hard time eating groups of cells. So this is a video that we shot actually as part of our outreach kit where we'll send you Snowflake yeast. If you're a high school or college student, you can play with them yourself.
I have it. It's right here. I wanted to ask you about
it. That's awesome. Well, I'll send it to anyone, to be honest. You don't have to be a high school, or you don't have to be a student. You could just be some dude in your basement. I'll send it to you anyway. So basically, yeah. So here you're looking at rotifers, and they're cool because they have a clear stomach, and they're filter feeders. You can see it's sort of feeding away here in this video loop. And we've given them red labeled single cell yeast and blue labeled snowflakes, and you can look at their gut. You can see their gut is almost entirely red. There's like one blue cell. It's kind of hard to see right here, but for the most part, it's eating the unis. So this could select for group formation as a way of avoiding being consumed by a predator. But we didn't use predators in our main yeast experiment, partly because, well, I'd say entirely because predators are they're a pain to work with. They tend to die on you. Their numbers change over time, so it's hard to control the strength of selection. It's hard to quantitatively modify the strength of selection. Let's say you want to increase or decrease it. How do you do that? They don't like the same media that yeast like. You know, the list goes on. Hard to keep the environment sterile. So instead, we use essentially a very simple physical phenomena of sedimentation. So we grow replicate populations in a test tube, which is shaken rapidly for basically for 24 hours, in which case the yeast are just growing, competing for sugar. Then at the end of the day, we take a random subsample, put it into a centrifuge tube, give it a light spin. Anything which makes it to the bottom has a chance of being passed on to the next tube. And if you don't make it to the bottom, you're killed. And it turns out that groups make it to the bottom much more efficiently than single cells, in the same way that if you throw a bowling ball into a lake, it's going to sink fast. But if you ground that bowling ball into dust, the little particles wouldn't sink very quickly at all. And in this first experiment, we basically found that snowflake yeast evolved in all 10 of our independent replicate populations within 2 months. And you can see kind of cool variation and density of these groups, their shape and size of cells, etc. Now, in general, there's 2 routes to multicellularity. This is true for any multicellular transition. You have an aggregative route where free-living single cells come together to form a group, and then they go off and do something multicellular and cool. This is what the cellular slime molds tend to do. So this is a little dictostelium group, which they forage as amoebae until they get starved. Then when they're starved, they aggregate into the slug, which then it runs around, finds a good spot, and differentiates into a stalked sporangia. Or you can have cells which stay together after division and form a group by dividing and remaining attached to their siblings. And this is, what the vast majority of multicellular taxa do. And there's a good evolutionary reason behind this. With coming together, you have within group genetic variation. Some cells are not, necessarily related to other cells in the group. And that means selection can still act at the cellular level. This is sort of most obvious when you're talking about the evolution of altruism. Why be altruistic within a body when, you could maybe gain additional reproductive opportunities by not being altruistic at the cell level and increasing in frequency within the group. So if you have a group that's genetically diverse, you have the potential for within-group evolution. That's not true if you have clonal groups. One of the building blocks of evolutionary processes is that there needs to be heritable variation for traits that affect fitness. And if you don't have that within a group, then you don't have problems within-group conflict. And that's why we only worry about cancer when we're old or we've been exposed to a lot of mutagens, because that builds up within-group genetic diversity, which then allows selection to act within your group. And that's not what we want for multicellularity. So it turns out that in yeast, group formation could have arisen through either mechanism. In fact, coming together is really common in yeast. It's called flocculation. If you enjoy lager beers, they result from flock-type yeast. And these yeast just basically produce an adhesive glycoprotein on their cell walls, which causes them to stick to other yeast. But that's not what happens with our snowflake yeast. Snowflake yeast basically have cells which form groups through staying together. In fact, you can kind of see these in this, I'm not sure how well this picture is coming across on your TV screen, on your computer monitors, but you see this kind of like branchy structure. We've stained this cluster here with a fluorescent cell wall stain. And turns out that daughter yeast cells are produced on one pole of the mother cell. So this is a good, let's consider this mother cell. It's produced these 3 daughters here. Can you guys see my mouse, by the way?
Yes.
Great. It's produced these 3 daughters here, and these very bright things are called bud scars. It's a strong chitinous link of cell wall material which holds on to daughter cells. So they basically, they form groups by not letting go of daughter cells once those daughter cells are mature, like they normally would, like they would normally let go of them, but they don't. And it also has this cool effect of giving you this branchy growth form, which is kind of like a coral or a tree growing as 3-dimensional clusters. When we look genetically, we find that this is actually due to a pretty simple change. If we look at gene expression differences between our yeast, we only see between like the unicellular ancestor and really early snowflake yeast, we only see a small number of changes in gene expression, and they're all related to things like this CTS1, which is an endochitinase, whose job it is to digest this mother-daughter chitin connection after mitosis is complete. So it's getting knocked down. Turns out that that's because there's a transcription factor called ACE2. ACE2's job is to turn on these endokinases, which digest that connection and allow daughter cells to float free. And that's getting mutations which knock out its function. And so, for example, we can actually do simple synthetic biology to show this is the case. This is our ancestor. It's got 2 functional copies of ACE2. If we knock them out with an antibiotic, just replace them with an antibiotic, we get snowflake yeast. And we can take experimentally evolved snowflake yeast, which got a natural mutation in the same gene, and we can repair that by taking this guy's ACE2 and sliding it in, and we bring them back to unicellularity. All right, so it's easy to get clumps of yeast, and in fact, in our 10 replicate populations, we have a bunch of independent mutations arising differentially within this gene, so it's a really common target. We see a lot of parallelism. Easy to get clumps, but are these clumps of yeast multicellular? I think it's in some ways sort of the big evolutionary question, right? What's an organism? What's an individual? How do you think about major transitions? Are these groups a major transition? What does it mean? So, I'm an evolutionary biologist, which means that I am required by the law of our Society for the Study of Evolution charter to have a Darwin quote in my talk. It's not really true, but may as well be true. So Darwin, though, thought about individuality, which is pretty cool. And this is when he was like voyaging on the Beagle, well before he wrote The Origin of Species, 20 years before. So he was seeing these colonial siphonophores in the ocean, and he wrote, each polypus, though closely united to its brethren, has a distinct mouth, body, and tentacula. Of these polypi in a large specimen, there must be many thousands. Yet we see that they act by one movement, that they have one central axis connected with a system of obscure circulation, and that the ova are produced in an organ distinct from the separate individuals. Well, may one be allowed to ask, what is an individual? Turns out that Steinbeck, this would be 112 years later, wrote a similar question, or a similar set of questions looking at similar organisms. There were colonies of pelagic tunicates which had taken the shape like the finger of a glove. Each member of the colony is an individual animal, but the colony is another individual animal, not at all like the sum of its individuals. Here are 2 animals and yet the same thing. Which is the animal, the colony or the individual? Why, it's 2 animals and they aren't alike any more than the cells of my body are like me. So, the fundamental idea.
Sorry to interrupt, but what are pelagic? What is that animal?
Yeah.
Can you quickly Google an image? That would be very helpful.
You want me to?
Yeah, if you could project it on the screen.
Sure. Yeah. All right.
Yeah, well, I guess the question I was wondering is because I know there are some weird things with polyps that like some of them are like superorganisms and others aren't. Yeah. So which ones were these guys referring to?
They were referring to these colonial ones, where essentially you have a bunch of repeated motifs on animals, like it's the same unit, right? Like this is homologous to one animal, but they're all connected together, and they clearly have sort of group level traits, which can't be explained by any single polyp. Like they have reproductive organs that are separate from all of these separate animals. Turns out that all these animals in the group are clone mates of each other. Here's another one, right? The Portuguese man of war, quote unquote, jellyfish. Turns out that the float, the feeding tentacles, the reproductive tentacles, the stomach are all developmentally derived from a different animal.
So all of those units, are they made by different zygotes, but they're all clonal?
They are made from, I think they are, yeah, they're different zygotes, but they're reproduced by early cloning.
Okay.
After like, you know, there's meiosis once, then you have like cloning causing you to get a bunch of little units which can separately develop.
So they're basically genetically identical, but there might be differences because of recombination effects?
I think they're genetically identical. Differences genetically are probably pretty minor. It's more like if you're a developmental biologist, it's like I can tell that these are, you know, basically animals, and yet they're all connected together. So what the hell's going on? Is this now an individual, or is it a part? You know, it's cool because it's kind of like you don't need a microscope to be able to see the sort of part-whole relationship. So it was very amenable to people like Darwin who are just scooping stuff up from the ocean and taking a look at it and trying to figure out what's going on.
Cool.
It's cool because it really does get at the fundamental idea of transitions and individuality. It's not quite the multicellular one, but you know, we don't have any Darwin quotes on multicellularity, so it'll have to stand in. Oops, dope. Sorry, guys. All right, so I want to go back to the criteria for multicellularity that I listed at the very beginning of my talk. We'll apply them to snowflake yeast. So snowflake yeast can consist of multiple cells that are physically attached. Check. Second, do they have a multicellular life history? So to get at that, we can just look at their life cycle. And here we're going to follow this one cluster. And it's going to pop off some propagules. I'm falsely coloring it. You know, propagules in green, parent in blue. And basically, there was a pair of twins that were liberated. They have this life cycle, which is an emergent property of physics, where they grow and hold on to their daughter cells. But at some point, they get too big, and the strength of sort of the cell-cell forces, the amount of force acting on cell-cell connections exceeds the tensile strength of a cell-cell connection, and you break a branch. And when you break a branch, a propagule floats free. So you have this life cycle that didn't evolve for a reason, but it's there, which I think is actually, that's really, to me, super exciting, because it shows how you get something very profound for free. We're going to talk about that more. And it turns out these propagules are functionally juvenile. By that I mean they grow for a while before they split. Once they get big enough and they begin to experience a lot of cell-cell forces, then they'll break off and pop off a propagule. This is just a graph showing size at reproduction, or sizes at reproduction. These are parents and these are juveniles. And you can see these size distributions don't really overlap very much. So turns out that, you know, newly produced propagules are almost always too small to reproduce until they grow. So that kind of gives you this juvenile stage. All right, so the next critical step is that adaptation takes place in the context of the multicellular group, or in multicellular level traits. And you know, we can go back to I would say the recent Darwin of major transitions, Maynard Smith and Zatmary, right? And they say that major transitions, like multicellularity, are major stages in the evolution of complexity that involve a change in the level of organization, and hence the level of selection. And I think Dave Queller, who is you know, a collaborator and spouse of Joan Strassman, who you just heard from a couple weeks ago, they've been working on this topic for a long time, and I think he has some of the clearest, cleanest writing on this. I just love this quote. He says, this is actually in a book review of, I believe, Maynard Smith and Zathmary's book. We designate something as an organism not because it is n steps up on the ladder of life, but because it is a consolidated unit of design. It is the focal point where lines of adaptation converge. It is where history has conspired to make between unit selection efficacious and within unit selection impotent. I think it's beautiful writing, and it sums up the evolutionary process. So we can kind of diagram this evolutionary process out. Cartoons for our yeast. If we start with the single-celled ancestor, selection favors the formation of groups. This could be because they're not being eaten by a predator, or because they can form groups which survive settling, whatever it is, they form groups. Now, once you have groups, if groups possess heritable variation in their group level phenotype, and selection acts on those group level traits, then you'll have corresponding adaptation in group level traits. And if that occurs, then they've made that sort of fundamental evolutionary shift to multicellular adaptation, where selection is acting on multicellular traits and the clusters are responding by multicellular adaptation. And if that's happening, then essentially, in my book, they've made the critical shift in evolutionary process, which is necessary for multicellularity. So in our experiments, we did select for fast settling, and we get the evolution of these clusters. And it turns out that clusters vary quite a bit in their multicellular phenotype, and that variation is very heritable. We've actually measured this variation for group size directly, and it's 0.84, which is really high. That's about as high as Daphnia's broad sense heritability. So then the question is, can selection act on those group level traits? And will they respond via group level adaptation? So to get at that, we did what's known as a divergent selection experiment, where we take a single population of snowflake yeast and either subject it to very fast settling selection. So strong settling selection, we only give them 5 minutes to get to the bottom of a test tube. And if they don't make it to the bottom in 5 minutes, they're killed. 15 or 25 minutes. This turns out to be pretty weak settling selection. 25 minutes is plenty of time for everyone to get to the bottom of the tube. So over 35 transfers, we see a response to selection. With strong settling selection, they evolve to sink faster, and that slacks off as we give them more time to get to the bottom. And the reason behind this is pretty obvious. In the strong selection lines, they evolve to get big. And in the weak selection lines, they evolve to get small. Keep in mind that these, in fact, do share a common ancestor, so that's a pretty big divergence in just a month of selection. And if we look at this in the context of this emergent multicellular life history, we see, changes, adaptive changes happening in this new multicellular life cycle. In the 5-minute line, those groups are evolving to delay fragmentation or delay reproduction till they're twice as large as their ancestor. Whereas in the 25-minute line, they're in fact evolving to fragment at a smaller size. So our snowflake yeast, and we have many more experiments playing with snowflake yeast adaptation, but snowflake yeast never failed to produce strong results to adapt rapidly to whatever multicellular selection you throw at them. They're really good multicellular individuals, Darwinian individuals, in the sense that they possess heritable variation for multicellular traits that affect fitness, and if you select on those traits, you'll get a response in selection. And I think that almost all of this arises for free as a side effect of geometry, and it's really cool. It's also really exciting to me because it's a very general argument about what it takes to sort of make a major transition happen. All right, so I want to walk you through the way that snowflake yeast grow. If we take a single cell, it's going to produce a daughter, and each of those will produce a daughter at the next time step, and they'll all reproduce at the next time step. And I've color-coded these generations. So this is the first cell, here's the second rung of cells, third rung of cells, fourth rung of cells, and finally the fifth one.
Do you get generation times that actually match up across the cluster as nicely as what you just described?
Yeah, in fact, I'll show you data on that in the next slide.
Cool.
With one caveat. But yeah, in any case, what's cool about this is that it actually fits the distribution you'd expect from Pascal's triangle, where there's one basal cell. This is the rung here represents the number of generations that have elapsed. So for the fourth generation guy, we'll go into the fourth rung here. It's one basal cell. It's got 4 daughters on it, 6 granddaughters, 4 great-granddaughters, and 1 great-great-granddaughter. So to Max's question, how well does this simple prediction fit what we actually see? So what we did is we made a really simple predictive model where you just count the number of cells in the group, and from that determine the number of doublings. And I should say that we started all of our experiments out with snowflake yeast that we had mechanically reduced to single cells. You can use, essentially, you can crush them between glass slides and break them apart. Then we get the distribution of cells expected if they're growing according to this simple snowflake yeast growth form. And then we plot the predicted distribution of numbers of cells based on the number of doublings they should have gone through against the observed number of cells at each position in the group. Now, the caveat that I mentioned was that for this experiment, we started with single cells that were allowed to go through, I guess, like 4 to 5 doublings. So there would be, is that right? 16 to 32 cells per group. So there's not a lot of diffusion limitation yet. So they're not, the inner cells aren't running out of food or oxygen. So given that caveat, we basically find a very nice correspondence between our predicted distribution of cells and our observed distribution of cells. The slope of these lines, this is for 5 different genotypes from different time points in the experiment. Slopes of these lines are all very similar, and none of them are different from 1. And there's some noise around here, and that's partly because we actually had to make the model account for fractions of cells because we didn't constrain it to even base 2 number of cells per group. So if there's, say, 17 cells, then the model would basically say, all right, cool. Well, we got the 16-cell body plan, and then I'm going to assign a 1/16th chance that you have an extra cell popping off of here. I guess a 4/16th chance that you have an extra cell popping off here, a 1/16th chance here, a 6/16th chance here. So basically that extra cell could go anywhere in the group. Now you have fractional cells. It doesn't really matter, but basically means that you'll never get a super tight fit because you can't have fractions of cells in real life. But in any case, take-home message is Snowflake yeast grow like they should. They have a very simple growth form. Remain attached to your daughter cell, and generally speaking, divide opposite from the pole from which you were produced. So it turns out that this growth form, this very simple growth form, which is a result of a single mutation, largely solves the problem of genetic conflict, which to many people is the hurdle you need to overcome in a transition to multicellularity. Because if you take this, oh, I'm sorry, I had my mouse on the wrong screen, guys. I apologize if I was pointing things out and you couldn't see what I was pointing to. So if you take this snowflake yeast and you choose any cell-cell connection to break, let's say I choose this cell right here. I'm going to break it from this cell right here. Well, okay, then this whole group floats away, and this whole group floats away, but this becomes a single-cell genetic bottleneck, and everything downstream of it is clonally related to it because they're derived mitotically. If this is a mutant cell, let's call it a hypothetical cheat, and I break it from its parent cooperator, well, now you've basically liberated an entirely cheating propagule, which can't survive because it has no one to cheat, from an entirely cooperative group, which is now rescued from cooperation, from cheating. Turns out that no matter where you break this, you're going to liberate an entirely clonal propagule of cooperators or an entirely clonal propagule of cheats. I could break this cell-cell connection here. Now we get a 4-cell cheat propagule. These guys would still be chimeric, but that's okay. The proportion of cooperators has increased. If I break this guy off from here, I get a 4-cell cooperative propagule. This is still chimeric. No problem. These guys are now completely freed from the problems of cheating. So you have this simple, structured growth form, which ensures that every single time you break apart, you're going to result in a clonal offspring, which either frees you from the problems of parasitism or cheating, or gives you a propagule of entirely clonal cheats, which is gonna do poorly. This is in conflict to an aggregative life cycle where it becomes very hard to get these kinds of bottlenecks in a life cycle because you basically can have cells that are free living kind of re-agglomerate into a group, and so it's hard to keep cheats out. Turns out that not only does this snowflake yeast life cycle, not only are these bottlenecks important for limiting the problem of cheats, but they're actually very important for increasing variance in cluster level traits. Now, I was making an evolutionary argument before that multicellularity happens when selection is acting on groups, and groups possess heritable variation for multicellular traits that affect fitness, which is, again, sort of the Darwinian algorithm, right? You need there to be variation. It's got to be heritable, and selection has to act on it. The rate at which adaptation takes place depends on essentially the ratio of between group variance to within group variance. In essence, that's what heritability is, right? How much does a genotype, how much between two different groups is there, how distinct are two different groups so that selection can act on those differences? If they're not very distinct, selection can't really see them very well. And how much, among a clone of cells, how much variation exists, right? If there's a lot of noise within a developmental body plan, then that doesn't bode well for selection being able to differentiate between different genotypes clearly. All right, so sorry, my throat is getting dry. So consider a beneficial mutant. This blue cell now is a beneficial mutant. These cells are twice as big as the ancestor. which in our experiments, that's actually a good thing. Bigger cells means bigger groups, which means faster settling. All right, no matter where you break this group apart, and you can do this, on your own screen, no matter where you break this group apart, you're going to get a uniclonal offspring. There's nowhere that you can break this 50-50 cluster where you're going to get a chimeric offspring.
And this is a formal consequence of the fact that cells are only connected to either their direct parents or their direct offspring.
Exactly. Very, very clearly said. All right. That's not true with aggregates. Here, most of the time, unless you're liberating, basically, this is a combinatorics problem. Most of the time, when you make offspring here, they are polyclonal. They are chimeric. You can actually formally analyze this and plug these different phenotypes of bigger or smaller cells into a simple physical model. Calculate, sort of terminal settling speed using Stokes' law calculations. And basically, if you have cells that are twice as big and the entire Snowflake yeast group is cells that are twice as big, you get settling speeds that are about 20% faster, or you're going to get things that are small, and they're going to be about 20% slower than this ancestor that's totally chimeric. For the aggregate group, most of the offspring you get have a 50-50 ratio, and you see very diminishing numbers of ways in which you can get propagules which give you biased genotype ratios, such that they're mostly large-celled or mostly small-celled. That's what these small filled circles represent. The filled circles are this sort of aggregative life cycle. So one of the nice things here, right, is that you can immediately see that Snowflake yeast, this simple body plan, maximizes the among cluster variance in fitness, such that you have things which settle very quickly or very slowly, and so selection can act on those multicellular fitness consequences. And because you have these regular bottlenecks in this life cycle, it means that every mutation which arises and results in its own group, because every mutation will eventually either result in its own group, clonal group, or go extinct, every mutation which arises is screened in terms of its multicellular fitness consequences. If it impacts their multicellular fitness, then selection has a chance to act on that, which is, I think, really cool. So getting back to Queller's quote about individuality, I think that this, Snowflake yeast demonstrate that simple geometry, which is the result of a single mutation, can conspire to make between-unit selection efficacious and within-unit selection impotent. You don't really need any magic developmental programs. You just need geometry. Any questions before I move on? Okay, I'll move on then. Cool. Going back. So the next question is, do we see adaptation occurring at the multicellular level? Do we see cellular specialization? I'm going to briefly talk about the evolution of programmed cell death in Snowflake yeast, which I think we're pretty confident now is a very simple early developmental mechanism for them. So our early Snowflake yeast are basically yeast cells stuck together. You don't see a lot going on outside of occasionally a dead cell or two. This is a cluster of cells stained with a green apoptotic stain or cellular programmed cell death stain and a red viability stain, and there's just one dead cell here and there's nothing apoptosing.
So the same population stain is not a stain for cells that are undergoing apoptosis now, but for cells that have transcription factors for it, or what's the, can you clarify that?
Yeah, great question. So the green stain is actually a reactive oxygen species stain, which is the sort of a precursor to programmed cell death in yeast.
Cool.
So these cells are full of ROS. This cell has a lot of ROS and is dead. In fact, many of these are. It's very common. All of these orange ones, and then these ones that are red are just dead, and we don't really know why. But they probably died from PCD in the past, and the ROS just washed out of the cell. Or they died for other reasons. We can't tell. So before I tell you sort of more about programmed cell death, I want to just give you a little primer on the evolutionary ecology of Snowflake yeast in terms of how to think about adaptation for them. Two things matter for their fitness. The first is their ability to reproduce. They are thrown into these tubes of media, where they have to compete fiercely for a fixed amount of food. And at the end of the day, basically, if they use that food faster than competitors, they have a fitness advantage. They are overrepresented. So the second thing is that they need to be able to get from the top of the tube to the bottom of the tube. Otherwise, they are killed during our settling selection assay, and that happens at the very end of the day after 24 hours. So in fact, these are just snowflake yeast from time 0, early snowflake yeast, and snowflake yeast that have adapted for, I don't know, 2 months. You can see differences in their average rate of settling. Now, it turns out that there are probably trade-offs between these two traits. If you want to be fast settling, then it pays to be big. But if you're big, nutrients don't diffuse in very quickly. So internal cells may not be dividing much, so you may grow slow. If you're small, you don't settle fast, but every cell has direct access to resources. And in fact, for our snowflake yeast, we see really amazing trade-offs. These are the first evolved snowflake yeast from 5 different lines. So we see this beautiful trade-off between growth rate and settling rate. And if we let these guys adapt for another 400 generations or so, we see something kind of cool, which is that 3 of them adapt by mainly evolving to settle faster, not grow faster. 2 of them, 2 of them do the opposite. They adapt to grow faster, not settle faster. So they're really doing different things here. And yet there's this beautiful parallel trade-off curve where they're basically pushing away from the origin. They're getting better at settling and growing as a whole, but they're doing it in different ways. So they're adapting around this trade-off, this fundamental trade-off, that it pays to be big, to sink fast, but it's costly because you grow slowly. Now, if you think about this from the context of one of our big-bodied snowflake yeast, if they divide symmetrically by splitting evenly in half, then they're always going to settle fast. They're also always going to grow slow. But they don't always need to be fast settling. They only need to be fast settling at the end of the day after 24 hours. And so if that's the case, then a more asymmetric division strategy might be better, because here they can produce small offspring, which themselves are not diffusion limited, and if they have sufficient time to grow, can grow to a large final size. So it gives them an advantage in this very periodic environment in which they live, where there's 24 hours between disturbance, where size is in fact a bad thing. You just want to be as small as possible to grow as fast as possible. At the end of 24 hours, you got to be big or you're killed. So a bunch of modeling and stuff suggests that asymmetric division is advantageous. And in fact, we see them evolving to be much more asymmetric through time. And the way that they get asymmetric is by having cells commit suicide. That's actually one way of we think we actually have some other ways in which they're doing it too. Work in progress. But when a cell commits suicide, it becomes a weak link in this cluster. And so if you have this cluster that's growing, the largest sort of strain on a cell-cell connection are those from the deep interior, where you have the largest amount of cells sort of pressing on other cells, generating tension. That tension is propagated down these chains of cells. Eventually, you have fragmentation close to the interior. With cells that commit suicide, they're a lot easier to break off. And the majority of propagules, once PCD is happening, have a dead cell at their base. And rather than breaking from the interior, you break from wherever the dead cell is, which is often closer to the exterior, giving you a smaller propagule. All right, well, is this process adaptive? Well, we are working on the formal, the formal sort of experiment, the definitive experiment, in which we essentially, we have candidate genes for programmed cell death, and if we add them to a strain which is large but doesn't have programmed cell death, or take one which evolved programmed cell death and repair those genes to the ancestral state, we can basically do these knock-in, knockout experiments to really show that the presence of PCD increases fitness. But in the meantime, we have all sorts of other experiments that suggest it's adaptive. For example, we can do mathematical models in which we invoke evolutionary algorithms to select on our digital snowflake yeast. I'm just pausing this little video. We have 2 co-evolving strains. They get mutations which affect reproductive asymmetry. So some cells die, which makes them reproduce more asymmetrically, and that affects growth rates. And so this has all of the trade-offs that you might imagine there are, right? That if you have some cells dying, then you're losing some cells from your reproductive pool, and so you grow a little slower. If you have some cells, if you're producing smaller offspring, then if they're produced near the end of the day, then they don't have very much time to grow before they run out of food, and so they actually lose during settling selection. So those are costs of programmed cell death. But programmed cell death gives you smaller propagules, which grow a lot faster. and parameterizing this with with data and simple physics when we don't have relevant data, in fact we see basically a coevolution between these two strains driving the evolution of a lot of programmed cell death. And this is actually really robust to the different assumptions of the model. You can throw in lots of different parameters and you get the same basic result. And this largely stems from kind of arms race dynamics in this, where if you're a little bit bigger and faster settling and a little bit faster growing than your competitor, then you clean the floor with them, and they can basically, you can kind of end up with these strains really driving each other to very high levels of programmed cell death. And we see a lot of parallelism. This is probably this and cell size, apoptosis and cell size, increased cell size, are the most parallel traits, and I guess increased cell number per group, the most parallel traits in our experiments. So a grad student of mine right now is doing an experiment with 20 replicate populations of snowflake yeast, and we're at week 24, and I think all 20 populations long ago evolved elevated programmed cell death. So it's incredibly parallel. It's a trait that seems to arise readily and is selected for in our populations. So this is not data from that experiment. This is from an older experiment, but we see a nice correlation between size, here described by settling rate. So just whatever, it's an assay for size, for settling speed, and percent apoptosis. So basically we see that bigger things have more programmed cell death. Now, this sort of explanation I've given you is that if you're big, you grow slow, you're under this, it's advantageous for you to have asymmetric division. Programmed cell death gets you there. But it's also possible that programmed cell death is not adaptive, but is just a side effect of size, because big clusters create a big diffusion gradient. Internal cells are stressed out. There's a lot of waste product buildup or low amount of O2 or food or whatever, and that causes them to undergo programmed cell death. So we can do a really cool simple experiment where we take early evolving snowflake yeast and snowflake yeast that have programmed cell death, and we can actually gene underlying group formation. We can repair that gene, bring them back to unicellularity, and see what their rate of programmed cell death is when they're single-celled versus multi-celled. And what's nice is that we basically find that the rate of programmed cell death doesn't change, that the 14 transfer guys, which haven't yet evolved programmed cell death, still have this very low background level. This is for unicellular yeast derived from snowflake yeast. The 60 transfer guys brought to unicellularity still have around 2%. So it's just basically, this shows that group structure doesn't play a role in programmed cell death.
Do you know what is triggering? Because it would seem very strange for a unicellular yeast to just kill itself.
Mm-hmm. So these unicellular yeast,
about what triggers it?
Yeah, sorry. These unicellular yeast they think they're still snowflakes. So they were evolved as snowflakes for 500 generations, and then we genetically engineered them to grow as single cells.
Yeah.
So they don't really know that they're not a snowflake anymore, and they're just doing whatever their genetic programs tell them to do, which in this case is 2% programmed cell death. Yeah, and we have candidate genes. There's a lot of mitochondrial, a lot of mutations which affect mitochondria in our snowflake yeast, and mitochondria are sort of the triggers of programmed cell death in yeast. And so we are in the actually I have a new postdoc that just started last week who first thing he's doing is going through and testing these mutations.
Cool.
All right. So to summarize the kind of key results of our main experiments, starting with a single-celled yeast, we did a very simple natural selection experiment in test tubes, selecting for fast sedimentation. And from that, we get snowflake yeast. And I'm saying natural selection here as opposed to artificial selection, only because the artificial selection, as the experimenter, we typically determine what the fitness payoffs are based on phenotype in an arbitrary way. Like you say, I'm going to screen things and pick the largest whatever. I'm going to screen maize and take the ones that have the largest number of kernels per year. Here, we didn't really know what was going to evolve. They could have taken different routes. They could have flocculated. They could have formed big cells. They could have done a lot of different things. But snowflake yeast is what they all settled upon. And once snowflake yeast evolved, selection shifts from acting on single cells to whole groups. because whole groups become a unit of selection. They either make it to the bottom of the tube and survive to grow another day, or they are transferred into the garbage can and killed. And snowflake yeast clusters readily respond to this group level selection. If we select for faster settling, we get the evolution of larger groups. And if we select for slower settling, we see the evolution of smaller groups. And one of the kind of neatest traits that we see is the evolution of a simple form of division of labor, where some cells give up their life in order for the group to reproduce. And this appears to be, it is incredibly parallel, and it appears to be adaptive. So one takeaway is that simple multicellularity, nothing as complex as a mushroom forming fungus or anything, but very simple multicellularity can evolve quickly. There's not a big temporal barrier there. It also suggests that, so the key to making this transition to multicellular level evolution, where you have things which grow as multicellular groups, and they have heritable variation for traits that affect multicellular fitness, and they respond to multicellular selection with multicellular adaptation, the key sort of evolutionary mojo behind a major transition, that appears to be pretty easy. And it has a lot to do, in my opinion, with these early life cycles, which have genetic bottlenecks and which allow there to be minimal genetic conflict because you don't have within group genetic diversity, and give you a life cycle and maximize between genotype variation, or between group / genotype variation, and minimize within group among clonal group variation. So in our case, we don't have a developmental pathway for giving you snowflake yeast. We have geometry, but it has the same outcome, which is that two snowflake yeasts of the same genotype are going to form groups that in fact look really similar because they have very simple rules for how they grow. Physics acts basically the same on each of them, so they end up doing kind of the same thing. We have a forthcoming paper on this topic in Filtrans B, which they're just putting the finishing touches on the special edition on innovations in evolution or in life. So keep an eye out for that if you want to learn more about life cycles. It's all about life cycles. All right, so in another sense, in a more astrobiology sense, our work suggests that if we can get simple, but I think honest to goodness, little multicellular critters evolving in test tubes in just 2 months, what limits the evolution of multicellularity on planets that contain trillions of cells and have geological timescales of hundreds of millions or billions of years for selection to play out? I honestly don't know how it, I don't know what the critical barrier would be if the evolutionary barriers aren't that high, and they don't appear to be that high. Well, all right, so, yeah, did you have a question?
Yeah, well, I mean, maybe this should be saved for when you finish, but could it be that, although these yeasts can evolve multicellularity quite quickly, that their evolvability beyond a fractal tree cluster is limited for other developmental reasons?
Yes. The answer to that is yes. Although, so I think, I wasn't planning on talking about this, and a lot of it's really new, so I probably shouldn't say too much just because we're probably years away from publication. But I think you get very different kinds of multicellularity with a hard, essentially a rigid body plan, one that plants and corals and snowflake yeast, and some algae exhibit, versus a soft body plan where things are actually allowed to move around. And you have things like animals where you can have cells move positions and really form cool dynamical structures. We're actually beginning to play around with both of those, and we're developing a yeast system which has essentially simple rules for how cells assort in space and time, which is pretty exciting. So that's part of it. But then again, plants have a pretty rigid body, and they've done amazing things. I think another large part of it has to do with how much developmental plasticity there is for individual cells. How many different ways can gene expression create different phenotypic states? And so that has to do with I think that's largely why eukaryotes have done so well with their multicellularity compared to prokaryotes. And so yeast aren't the best model for that. They don't have very complex genetic architecture. They don't have a lot of microRNAs and alternative regulation, or opportunities for variable regulation. Yeah, so they're not the best organism in that way. But if you took snowflake yeast or something which forms soft mobile bodies where cells can move around and form embryos, and you did that with something that was a little bit more complex, and then you put them through similar selection for size, I think you would get complexity, because in my sort of experience with the system, one of the things which really drives increased complexity is just size. If you select on size, you can't just make a big ball of undifferentiated cells very easily, because they end up really running out of resources and stuff. They grow terribly. And so you end up getting all sorts of compensatory mutations to make them better at being big, make them not suck at being big. And that begins to build in structural complexity and developmental complexity, and you begin really laying the groundwork for the evolution of development, simply as a way of compensating for the stresses associated with being large. Physical stresses, you just have to solve physics problems to get big if you don't have an evolutionary history of it, as well as evolutionary problems that if you're big, you often are really bad at growing, and so you need some way of overcoming those constraints, and they'll often do that by getting more complex. And then if you have size, a life cycle, and the ability to have simple development where cells are doing different things at different places in the group, well, now you have the toolkit to really adapt as a multicellular critter to all sorts of different ecological niches. So I still think this problem stands in the sense of, if you have cells, why wouldn't you have multicellularity? It may still be very simple. It may be, cyanobacterial simple, but still, you should be able to get that. You guys want to see a few ongoing projects from the lab?
Absolutely.
Cool. So one of the things that we see, we're very interested in multicellular adaptation, and taking these guys down a long road and seeing what happens. So if we select for faster and faster settling, we see some things happening very consistently. This is days of evolution down here. Notice that it's a log axis. This is settling speed, so we can actually measure the settling velocity of clusters, individual clusters in media. And it goes up. They settle faster. No surprise there. Now, they do this in a number of ways. First of all, I should say at every time point, we end up packing more cells into each cluster, which is not trivial to do, turns out. We also see one-off changes in cell size and shape. So like in between these two time points, we see larger, more elongate cells. And that actually allows them to, we have a paper that we're in revision for right now, in which we're really diving into this phenomena of the physics behind how cellular elongation affects packing density and therefore group size. And it looks like elongation plays a large role in reducing the effective packing fraction of a group, so spreading things out a little bit more, which allows them to get a lot larger before they, it makes them much more tough in the sense that they can tolerate a lot more energy on those cellular bonds before they break one. And we also see our snowflake yeast evolving to be more hydrodynamic and settling more efficiently by having more spherical groups and having fewer of these branches hanging out into the media, acting as a drag. So our general approach to understanding these traits, like how do you get larger, more elongated cells, what's going on with programmed cell death, how do you get more spherical clusters, is to develop three-dimensional models, which are getting better and better, and actually getting to the point now where they're making predictions which can be tested empirically, and they are super good. Like, we can plug in a few key parameters about cell size and shape, and we get back data which falls directly in line with experiments for, say, how large the cluster should get before it breaks under its own sort of forces. It's pretty cool. We're tying in these physical models, and this is with my collaborator Peter Juncker, who's a soft matter physicist. We're tying in physical models with evolutionary models to see how changes in traits affect the physics of groups and how that affects selection acting on group level traits. And these evolutionary models are a collaboration between myself and Eric Libby, who's a postdoc at the Santa Fe Institute, soon to be starting his own position at Umeå in Sweden. And then we are basically doing genetics experiments to find the genes underlying these traits and go really from genes to physics to multicellular emergent properties to fitness, which is pretty cool. It's all part of essentially, yeah, this heading of understanding multicellular adaptation. So this is data from Peter Junker's group, in which we're looking at how early pretty spherical cell yeast and later more elongate cell yeast, how these guys are just much tougher to physical stress. So what we did is we tortured snowflake yeast by breaking them apart with an atomic force microscope. So we can actually compress them with an atomic force microscope and measure the amount of force required before they fragment. And we looked at that breakage energy input requirement as a function of their size. These are two different genotypes. The blue is this kind of roundy guy here, and the red is this more elongate guy here. And it turns out that these circles are their spontaneous, basically this is the size distribution, this is the size maximum to which they grow when they're growing unconfined. So you just let them grow in a test tube, and this is how big they get. These regressions here show that when they're small, if you add you add some force, it takes a lot of force when these guys are small to break them apart. But as they get bigger, they get much more brittle. It doesn't take very much force at all when they're just a little bit bigger, right? Going from 27 to 32 microns in diameter. And then you barely touch them and they break apart. And if you don't touch them at all and let them get big enough, then they'll just break apart under their own stress force, which is pretty cool. These more elongate guys, you can see that they have a much longer, slower slope here, meaning that they can get a lot bigger, but B, they're also much less sensitive to increased size before they spontaneously fracture, and this is their spontaneous fracture size. So by making cells more elongate, they actually evolve, they change their packing density, which has these cool and well understood physical consequences for the materials property of a snowflake yeast cluster. And interestingly enough, physicists figured out these basic rules for packing with Kepler 400 years ago. It took human physicists over 1,000 years to figure out basic rules of packing and how ellipses pack differentially from spheres. Same rules that snowflake yeast figured out in just a couple of weeks. But there were millions of them working on the problem. You can imagine that there's a huge payoff associated with getting more elongate for a snowflake yeast. We're also doing experimental evolution with crushing. So we're actually using this thing called a universal testing machine to deliver a set amount of force to a population of snowflake yeast, and then we're basically selecting for settling after they've been crushed. And if you evolve them with compression, this is the before crushing and after crushing distributions, and it doesn't change that much. If you evolve them without compression, man, they get very sensitive. If you compress them, they fragment like crazy. So we can evolve tougher snowflake yeast as well. A grad student in my lab has developed a really nice system for looking at the differences between flocculation or aggregation, that evolutionary foil to snowflake yeast that I keep talking about is like the bad way to be multicellular, and snowflake yeast. So in fact, these are just two different labeled genotypes of snowflake yeast. You can see they're clonal, either all green or all red, and the flocks aggregate and are well mixed. And what's nice is that she created these synthetically. So these flocks are derived from the same unicellular ancestor that the snowflakes are. Here we added a FLO1 gene, and here we removed the ACE2 gene. Those are the only differences between these two populations. And we previously had done a preliminary experiment where we took flock yeast from nature, which already settle fast, so they're pre-adapted to our environment, and we just selected on to continue settling faster. And we thought that they would just become more flocky. They already have a way to get to the bottom. But that's not what happened. Snowflake yeast basically evolved within the flock populations and took them over and drove the flock to extinction. So to kind of dig into that, using this system of synthetic flock and synthetic snowflakes, my grad student Jennifer Pence did some kind of cool social evolution experiments. And for this, we had to actually develop a new way of measuring settling speed, because flocker, turns out they're a huge pain to work with. It's kind of like that I think it's the Heisenberg uncertainty principle in physics, although if a physicist watches this and I'm using it wrong, email me. But basically, the whole thing of you cannot measure something without affecting its state. Turns out that's true for flocks. If you want to measure, say, how large they are, you want to put them under the microscope or pass them through a flow cytometer, which is the way that we normally measure snowflake yeast, you totally change the game. If you put them under a microscope, well, they begin to flocculate on the cover slip, and that's very different than how they would flocculate in a test tube. It turns out that flock yeast form groups as they're in the process of settling out of solution. So there's no way to say what their size is because it's constantly changing. So really, we had to develop a new way to measure size/settling speed that didn't rely on actually measuring size directly. So what we ended up doing was videotaping populations of flock and snowflake yeast as they're settling. So you can see that flocks settle way faster than snowflake yeast. They just form these big clumps and sink out of solution fast. The snowflake yeast, you can see it clearing up here, but it's a lot slower. This is 20 seconds compressed down into 10 seconds maybe of the video clip. This is 5 minutes compressed down into one run of the video clip. Huge difference in absolute rate. And then we basically just developed some image analysis software in which we could see how that biomass was changing throughout the height of the cuvette. And from that, we can fit some mathematical models to get a characteristic settling rate for the population. And we do that with lots of replicates of different populations or genotypes to get how fast they settle. It actually works really well, but it's kind of a hassle. But it's okay. It's worth doing since it works. Now, here's the surprise, right? I already showed you data suggesting that snowflake yeast evolved from within populations of flock and then drive flock to extinction. And this is for wild flocking yeast. So what's surprising is that in our experiments, the synthetic flock yeast appear to be superior. During growth, right, if we compete them at a 50-50 ratio just for growth, it turns out that the flock grows about 35% faster than the snowflake. That's because they're mostly single cell. They don't have these problems of diffusion. They get to form groups at the very end of the day when it's optimal. So growth, it seems like it'd be great to be a flock. What about settling? Well, I already showed you a video showing that flocks settle like 4 times faster than snowflakes. So if flock are better at growing than snowflakes, and they're better at settling than snowflakes, then why would snowflakes win? Well, turns out snowflakes act as cheats in chimeric groups. So this is a cluster of fluorescently labeled flock and non-fluorescently labeled snowflakes. So these gray large blobs are snowflakes, and these green fluorescent things are flock. And it turns out that when you mix flock and snowflake together, snowflakes get into the flock groups. They get into the flocks. And what's cool is that single-celled yeast that are not flocky, so the snowflakes are not flocculent. Single-celled versions of this yeast, they don't get into the flocks. They're excluded. But snowflakes get in. And not only do they get in better than the flocks themselves. So we can do an experiment where we start out a bunch of populations with different starting frequencies of snowflake yeast. And then we can ask what the competitive success of the snowflake yeast are, which is essentially like looking at an invasion experiment where we start out with 1% snowflake yeast and then ask, how does its fitness vary as a function of its frequency as it proceeds to invade? So when snowflake yeast are really rare, this is the line of parity, where flock and snowflake yeast are equally fit. When they're really rare, like 1%, they have a 2.8-fold relative fitness increase over their flock competitors. And basically this is because a bunch of flock cells don't make it into a flock. A bunch of flock cells are just single-celled and they didn't get into the flock. But pretty much every single snowflake yeast in the population makes it into a flock. So they have really high survival during settling selection. If we increase their frequency, then this in fact goes up more. And we think this has to do with essentially the snowflake yeast increasing the settling speed of their groups because they add so much biomass to their groups that they're actually playing this ecological role where the flock themselves gain a short-term advantage associated with having snowflake yeast in the group. The snowflake yeast gain advantage because they're in these flocks. They form these big, fast-settling groups, and it turns out that it's true. These chimeric groups settle much faster than pure flock, which is kind of amazing. But then as they increase in frequency, then those differences, then almost every flock has snowflake yeast in it. So the between flock differences begin to diminish, you get all the way down to the point of parity here. But it turns out that at 80%, you begin having so many snowflake yeast in the population that they interfere with flocculation, and the flocks stop forming. When the flocks stop forming, now it's snowflake yeast competing against unis. So these are flocks, chimeric flocks from this regime here. Here you have snowflake yeast competing against a unicellular flock, and that's just, you know, that's it's like Game of Thrones last night. It's like having a dragon come over and burn away your flock competitors, because single-cell flock don't hold a candle to a snowflake yeast cluster. So there's this kind of two-phase thing where snowflake yeast essentially exploit the aggregative mechanism of flock. They take advantage of the fact that flock can't really keep them out of their groups. And then once they've driven flock to a low enough frequency, they just mop up what's left over by competing against single-cell yeast. So I don't have the data in here, unfortunately, but Jen Pence, my grad student, is doing other really cool work. She's initiated 20 replicate populations of these synthetic snowflake yeast and 20 replicate populations of this synthetic flock. She's evolved all 20 populations for 24 transfers. Every 8 transfers, we've sequenced all of these populations, so all 40 populations. And we are looking at the genetic dynamics, the genetic dynamics of adaptation in flock versus snowflake, and looking at the sort of tempo and mode of adaptation. Are snowflake yeast more able to respond to group selection than flock, which are chimeric, and a bunch of other things. It's a pretty cool system that she's developed. Another student has been looking at sort of emergent, almost superorganismality. Can whole snowflake yeast clusters evolve to cooperate with each other? So she put snowflake yeast under settling selection that was so strong that individual clusters couldn't make it to the bottom. And what do? They evolved to produce these proteinaceous filaments which bind together whole groups. These are whole snowflake yeast clusters. So this is a large image. And these kind of metagroups are able to make it to the bottom of the tube and survive when individual clusters can't. And this is actually a repeatable phenomenon. This happens not every single time, but most of the time. And we think it's basically a consequence of programmed cell death, that we have very high rates of programmed cell death in these guys that evolved from these filaments. And these filaments are actually just internal cytoplasmic goo, DNA, proteins, and it just basically forms this population level benefit of forming these filaments, which are cheatable, because if you're a non-filament forming strain, you can get in at the same frequency, and you have pretty crazy evolutionary dynamics, where in fact the frequency of these filaments oscillates through time, and we think there may be some social evolution going on there. But it again kind of suggests or demonstrates how easily cooperative dynamics can be used to respond to very challenging environments, which could potentially precipitate a change in the level of selection. Here you're selecting on groups of groups, which is essentially what we did with the snowflake yeast, but now it's a group of snowflake yeast, where each snowflake yeast is a group of cells. So you begin to see this sort of nested hierarchy building up, even in very short-term experiments. And that's a filament in a test tube.
That's cute.
All right, I got like one more vignette here, or maybe 2, and then I'm done. I know this talk's been going on really long, but I was talking to Max, and he's like, "We have 2 hours," and I was like, "Oh, that's good. I will have all sorts of cool stuff." So I put way more stuff than my normal talk, and I'm going a lot slower than I normally would, which is kind of nice. So we're very interested in involving multicellularity in other systems. So in collaboration with Matt Herron, who's actually here at Georgia Tech and on the job market, he's really led the system. He's a Volvox guy. We're selecting on Chlamydomonas, which is a single-celled alga, for group formation, and we want to get to a Volvox. I don't think that'll ever happen, but we'll see how far we can get. So he did this experiment with 50 weekly transfers of a single chlamydia cross. 5 of these were transferred with single-celled predators, so paramecium. 3 of them were transferred without paramecium, so these are unicellular controls. 2 of these evolved to form pretty groups, and these guys stayed unicellular. So B2 and B5 formed groups. And it turns out that these early multicellular sort of forms that we see evolving in these chlamydia populations really closely resemble extant Volocene algae. So these These are actually Volvocene algae isolated from lakes, multicellular, bona fide multicellular algae. You can see this extracellular membrane, multiple cells. We see something very similar happening in our experimental populations. This is just a little bit of data showing the size of these different groups. So the controls are down here. We have some, some lineages from the multicellular lines, which, so, we're looking at multiple isolates. That's what this dash is. B5 is like the B5 line, isolate 6. Some of these are pretty unicellular, but then some of them became pretty multicellular. This is the average number of cells at the point in their life cycle when they were the largest. And this is sort of a life cycle analysis looking at the size of propagules. So many of our strains basically did evolve to form, to reproduce with propagules that are like 8 cells on average. This would be 4 cells, 8 cells, 16 cells. So they have pretty large offspring, and they in fact encompass several distinct life cycles. So we have things which evolved to form groups encompassed by an extracellular matrix, and they released single cell propagules pretty much exclusively. Or on the other extreme, things which form groups encompassed by an extracellular matrix, and they released 4 to 8 cell groups and almost never single cells. So I'm going to show you a few videos of what these things look like. So this is a unicellular control. I hope the frame rate's up to snuff. But this is kind of a boring one. It's basically, despite the high frame rate, it's just so it looks like popcorn where the clammy settles down, divides a few times. They do this cool palentomic mode of growth where the cells get huge and then they undergo several successive divisions. So that's what's happening here. They're dividing a few times and then single cells pop out and swim away. This is the classic clammy life cycle. So, so this guy, let's go back to here. This guy is one of the lines which forms groups, but when they reproduce, they basically release single cell propagules, which then swim away and form new groups. That's pretty cool. So these, again, have a single cell genetic bottleneck during development, which gives you all of those cool things that I was talking about before, minimizing the potential for within-group conflict and maximizing between-group differences in multicellular traits, which allow selection to act on emergent multicellular properties quite easily. And this one is these ones get the biggest. These are super multicellular, and they pretty much obligately have multicelled offspring when they produce propagules. They're also by far the slowest growing in our populations. But yeah, you could imagine if you're a little protist, you'd have a really hard time taking a munch out of these guys. They're just really big. All right. So I'm going to finish up here, but I just want to mention that while multicellularity is the main thing that our lab does, it's not really, my background is in social evolution, and so we're working on other things too, mainly experiments with spatial dynamics of microbial populations. And so we're doing a lot of stuff on Petri plates with bacteria, which duel with these. They basically kill each other by stabbing with antibiotic-tipped harpoons, and that creates phase separation. This is a simulation of a coffee ring effect, which is a physical phenomenon which structures microbial populations. And then we're working on bed hedging as well. This is all from a theory perspective right now. But we've got a couple papers submitted in a press on that topic. I'm happy to talk about social dynamics and spatial dynamics of microbes, or bed hedging if you guys want, or we can talk about multicellularity. I'll just skip this phase separation stuff. And with that, I'd like to thank all my collaborators, and there are many more than this, but these are the sort of often the people that I talk to the most, and the people that have actually done the work, people in my lab. Here they are being a snowflake cluster. It's growing, and then a propagule is going to separate right over here. Yeah, propagule separation. That's my first grad student, so she unfortunately has to apoptose. It's very sad for her. All right, bad joke. That's it. I'm going to stop screen sharing and take some questions. Thanks, guys.
Thank you so much. So I want to start with this. Can you tell me a little bit more about this yeast kit and what I can do with it?
Yeah. Basically, there's two things you can do with that. You can grow your own snowflake yeast, which is kind of fun, so you can play around with them. You can, we included a strain, in fact, that is very prone to evolving snowflake yeast. So it's heterozygous for ACE2. So all it needs is so it's unicellular because it's got one functional copy of ACE2, but one defective copy. And if you get what's known as gene conversion, which is just you copy chromosome, the defective copy from one chromosome to the other one, then you'll get a snowflake yeast. So you basically, if you grow those unicellular yeast in like 2 days, you're gonna have snowflakes popping up because of gene conversion. And then selection can act on those and increase their frequency. And so if you want to do experimental evolution of unicellularity to snowflakes, you can do that in your class in less than a week, which is pretty cool. The other thing you can do is, I think that's just YPD, growth media. The other thing is we included snowflake yeast and unis that were fixed and labeled red or blue. And so if you order rotifers from like Carolina Biological Supply, for example, then you can basically do these experiments where you sort of do the ecology experiment, like how would a predator eat single cells versus multi? And you can watch them feeding, you can look in their gut. Whenever I do outreach events, I take a microscope and I take these yeast, and people love it because first of all, those rotifers are super cool. When I've done like high school class outreach, I had like a 15-year-old like making up a metal song about them. I think we used one called Stentor, and he was like, Stentor, and I was like, that's actually a pretty good song about Stentor. I mean, that's like the most metal sounding protist.
What's this bit?
Those are depression slides. So you can look at them under a microscope without flattening them.
Oh wow, this is so cool. I wish I had a microscope.
Yeah, you can. Yeah, you could probably make one out of a cell phone. There's all these DIY cell phone microscope with like a glass bead.
Yeah.
Yeah, like a Van Leeuwenhoek microscope.
Wow,
a depression slide is a move on the guitar and a metal riff, right?
Whoa!
Sorry.
Yeah, that's good. That's good. Or it describes my psychological state after listening to a lot of metal. Total depression slide, man. Not my genre. Give me some Simon and Garfunkel.
Okay. So now I'm going to ask you a more theoretical question.
Yeah.
So Durkheim wrote this book, right? I think this was like the first book that he wrote, this foundational sociology book on suicide, where he points out that like suicide is like the social problem that we need to like. I can't paraphrase Durkheim. I've never read the book, but it strikes me as really interesting. And like my knowledge is not perfect, but I believe that the only animals that commit suicide on the regular are humans, and then maybe eusocial insects like bees that are killing themselves for their colony. So I don't know if that's true, but certainly with human there's something really evocative about apoptosis being the critical mutation, sacrificing your life for the good of the group. And I was wondering what you think the role is for suicide more generally in terms of these transitions, is it a coincidence that it's apoptosis that is stabilizing these structures, or is it more than that?
That's a really good question. I think you're very perceptive when you say that suicide is something you typically see with really social organisms, because in order to sort of make it evolutionarily advantageous, you need to invoke some form of kin selection or multi-level selection in order for suicide to actually pay off and be better than no suicide, in order to increase the frequency of a trait which causes suicide in the population. So parental care is a case where I think it's very generalized that you're going to see a lot of suicide. I'm a chicken owner, and I have seen my mother hen fly into the jaws of a dog to keep it away from her chicks, which is like, chickens aren't fighters. If you have ever had chickens, they're chicken, right? Like that's a term because they're afraid, and yet she's willing to like really risk death for her chicks. I guess maybe you wouldn't call it suicide, but from a biological perspective, it's taking an active step which results at least some high some fraction of the time in your own death. So yeah, but it makes sense because of inclusive fitness, right? It makes sense to do so if you're going to keep your chicks from being eaten or save a sibling or something like that, right? It makes sense in a multi-level, in a major transition perspective even more because of the exceedingly high relationship among parts in an organism. I mean, you can model it two different ways. You can think of, your cell that undergoes programmed cell death, doing so to help its sibs that are also in your body, or doing so to help its sibs that are even more importantly in your, future reproductive propagules. And it turns out that in a lot of multicellular organisms, apoptosis is very common. So when you get a sunburn, it's not because you actually got burned, it's because you had a lot of DNA damage, and those skin cells then commit programmed cell death, and that programmed cell death is what causes the burn. In our case, I think that programmed cell death is one of those things that there's a lot of ways to get it. So it's a very easily accessed evolutionary state for snowflake yeast. A lot of ways for a cell to freak out and die. And so it's mutationally, it's a rich target. And then because of this kind of periodic environment in which they live, it becomes like something which is pretty beneficial. That if you're big, it's actually good not to break in the middle, but break into smaller pieces from your body. And what's cool is that you mentioned that it's important for the stability of the transition. And in fact, it's really important for the stability in another way, which is something that we've termed ratcheting. So imagine that you take an early snowflake yeast and it's only been adapting for a week and you revert it to unicellularity. What would the fitness of the unicellular revertant be? Basically the same as the ancestor. It might have had a few mutations, but like it's pretty much the same as the ancestor. If you let it run out for 500 generations, and now you have bigger cells and programmed cell death, and you revert it. Well, we've measured this. The fitness of those revertants is lower than the ancestor because now they're single-celled yeast that are killing themselves at 2% of the time. That's a bad strategy. They're not growing fast. They have these bigger cells that actually grow slower, which are great if you're in a big group because it increases survival. But if you get rid of that survival benefit, then they're growing slower. And so those traits act kind of like Muller's ratchet in sex. It acts as a ratchet that if you accumulate enough of them, means that the reverse transition becomes very hard to do. The terminal endpoint of that is basically what is what we have, which is that if you tried to scrape your cells off, they can't survive outside of your body. And so, except in extreme circumstances like a laboratory. But we basically are terminally differentiated, which is basically the endpoint of ratcheting. But ecologically, if you're liberated individual cells, let's say you have a mutation which brings you back to unicellularity, and you're half as fit as your ancestor, then you don't stand a chance of competing against the other single-cell things that are still there. You're basically stuck being multicellular. And that has really interesting implications for the evolution of increased complexity because it means that in order to adapt to future environments, rather than switching back and forth between multicellularity and single cellularity, if the environment is variable, you're basically stuck being multicellular, and you've got to figure out a multicelled solution to that problem, which I think that's pretty cool. It sort of gets at one of the reasons that we see so much nested hierarchy within life, which is that in many ways it is easier to go up than to go down.
That's super interesting.
Yeah, right? Yeah, I love that stuff.
I've got more questions.
This is the reason to be a professor, is like all day long I just get to think about and do the things I think are cool. That is a great job.
I have some questions. I'm kind of curious so first of all, I want to try and connect this a little bit to Queller and Strassman's definition of organismality or what an organism is. And it seems like your research does have a very tangible or specific way of kind of backing up that definition of what is it, high cooperation and low conflict, and the consolidated bundle of adaptations making whatever. So I'm sort of curious of like how you think that this research does enforce that definition or doesn't, and also why yeast, like I don't know, this is maybe a separate question, but how is it that you found yeast, or I don't know, maybe yeast is typically studied in this type of field, but I think you mentioned at the end of your talk another similar research with algae, I believe. And I'm just kind of curious about why yeast and how other things like maybe algae also support this kind of definition of what an organism is.
Yeah, no, great question. So in terms of Queller and Strassman, it's kind of interesting. Like when you wade into the philosophy on this, it seems like there's kind of there's sort of two different. I think a lot of people use the term organism and individual interchangeably, and some people have been kind of trying to hold those two concepts apart. And I think, and so the way I tend to think of it is that organisms, and this is kind of borrowing Peter Godfrey Smith's ideas, which I, of course, take Queller and Strassman and a bunch of other people's stuff kind of into the same account as well. But an organism is something which is essentially a functionally entwined entity. So like a great example that Peter Godfrey Smith likes to use is a squid, a bobtail squid, and a Vibrio fisheri, which is fluorescent and inside its lux organ, or its light organs, right? So they're an organism because they work together and they're highly cooperative and they're functionally entrained. But they are not an individual because they're not an evolutionary unit, right? So if you define something as a Darwinian individual is a participant in an evolutionary process, then the squid is an individual on its own right, and the Vibrio is an individual in its own right. But because they basically have this short-term ecological association where the squid flushes out the Vibrio every day and takes new ones into its light organs, their coevolution is pretty minimal. And basically, they're not like a lichen, which is like maybe a more persistent association of a photosymbiont and a fungus, which is both an organism and a Darwinian individual, because selection is acting on the symbiosis itself, and they're being co-inherited in propagules and stuff. Or us with mitochondria, in which we're very much part of the same individual. And so the way I tend to think of it is that organisms essentially fit the Queller-Strassman definition of high cooperation, low conflict, functional integration. Whereas individuals are essentially the way I describe things of, do you have a basically heritable variation or traits that affect fitness? Is selection acting on those? And as a result, are you seeing adaptation? So it's very much an evolutionary, as a description of the evolutionary process. And you're asking, what is the focal point of adaptation? What is the focal point of selection? And those two things aren't always, they're usually the same for the kinds of things that we see with our eyes. Trees, for animals, they're the same. But when you really kind of get down into these gray zones, they're not necessarily the same. So I think Queller and Strassman are, I think they're largely, I don't think there's anything wrong with their work, obviously. I really like that paper. But the reason that I don't tend to talk about organisms as much as individuals is because I think it's possible to have higher level individuality, say, really without necessarily cooperation or conflict playing a large role. I think usually you will have cooperation and conflict, but I don't think that that's necessarily the defining trait. I think the defining trait is the evolutionary process. And cooperation and conflict are things which arise in the context of the evolutionary process and basically make the organism what it is. But to me, that's a result of evolution. And so it's the process of how is the evolutionary process playing out. That's the fundamental thing. And so that's why I like to kind of focus on, just because like normally, so for most organisms, you can't really observe that process. You can get an idea of where adaptation, you can get an idea of the process because you can look at traits that have evolved. And if I say, look, this organism has a bunch of cells that are being altruistic, and that appears to be adapted for the group, then you're pretty sure that cells aren't the focus of selection, right? It's hard to explain it if they're a society of cellular individuals. But it's easy to explain it if the cells are a part of the multicellular individual. So, if looking at traits often gives you insight into the evolutionary process, but if you can study the process directly, then why not? because it's rare that you can. To me, that's really the fundamental part.
So that's where the yeast comes in, right? I mean, where else can you study the evolution on that level?
Yeah, I mean, you need a model organism that you can grow under controlled circumstances, and you can know a lot about them. And one of the nice things about yeast is that you can freeze them, so I can actually go back to the actual ancestors of anything extant. It's easy to modify them genetically, so I can take genes that evolved in the future and put them into the past, or take genes that evolved in the future, repair them to what they were like in the past. You can do all sorts of great experiments with them that would be really hard in other organisms.
So yeast is basically...
Go ahead.
So yeast is basically like time travel, is what you're saying.
Yeah. Freezing yeast is time travel, for sure. You can freeze other organisms too, and we chose yeast really pragmatically. It's just an easy organism to work with. The downside of that is that the upside is there's a huge community of people studying yeast. So much is known about them. Any method that you want, you can do in yeast. Like, oh, I'm going to do CRISPR. Okay. Who's got a CRISPR system in yeast? This guy. Can I have it? Sure. It's great. It's easy. Downside is that people really expect you to do a lot of cell biology because you can. And if that's not really your main interest, then it can be kind of a drag. It can be a lot of like, oh, we would publish this in Nature, but you don't have enough cell biology. And if you had an organism that didn't have as many resources, they'd be like, well, we understand why you didn't do all the cell biology. So it opens doors, but then people kind of want you to go through all those doors, even if that's not the goal of that specific project. They kind of want you to go. They want you to do all the bells and whistles for every paper, which is that's the downside.
That sounds like a state of affairs that is not particularly unique to yeast research, although I can definitely see why it'd be acute there.
It's not unique to yeast research. That's right. But if I was working on some marine, single-celled eukaryote, which is a close relative of animals and didn't have a genome, I could get away with doing a really different set of experiments than I can get away with yeast, for example. Yeah. No one's going to expect you to nail the SNP and then do single-cell transcription profiling.
Yeah.
Because no one's ever done it before. But yeast, look at these guys. Their entire lab spent 4 years making this process work, so you should do that kind of thing. That's my snarky take on it. But no, overall, it's a good thing because so many things are possible, and I think that's a great thing.
Well, and your evidence is just so strong. Like, I read your first paper, and I was like, holy crap! That you got this so reliably and so quickly. It's kind of unprecedented for artificial selection experiments, or even if it's not, it's just I'm amazed. Like, it seems like you kind of lucked into this really, really fruitful. But I guess it wasn't luck. Like, you chose it very specifically.
Well, no, I think there's a lot of luck involved. I honestly, when I started the experiment, I thought we'd get flock because I didn't know about Snowflake yeast. I thought, well, we know that yeast flocculate, so I'll probably select for some flock. Actually, honestly, going back to Queller and Strassman, I had been kicking around the idea of doing this with Mike Travisano, my postdoc advisor, back when I was still a grad student. So I started this project as a side project when I was like a 5th year PhD student. Funny timing to start a side project. Maybe 4th year, towards the end of my PhD. And we'd been like, wouldn't it be cool to evolve multicellularity? But it was just a chat, and nothing came of it. And then Dave Queller came to the philosophy department at Minnesota and gave a talk on organismality, the paper that you read, the 2009 one. I'm assuming that's the one you read. Yep. He gave his talk on that, and the flock thing, because flock is one of the examples of a little bit of cooperation, a little bit of conflict. Okay, it's kind of organismal. And I was like, dang, let's do this in yeast. Let's get flock. Let's select on flock. And so, we had some yeast in the lab, and it turns out to be the strain that we used, which is a mostly wild strain, still highly outbred, doesn't have any knockouts or anything. And first thing we tried was essentially what they did in the flock paper, which is put them in an environment where there's an antifungal or an antibiotic. And if the, if the flock, the cells on the outside of the flock die, but the inside survives. But you can imagine what happens if you try that to a population of single-celled yeast that don't have standing genetic variation for group formation. You do exactly what those antibiotics are designed to do, which is just kill everything. So I just killed everything. It was like, we need a solution that isn't quite as harsh. And so that's where settling selection came in. It was a much more gradual thing that even if there are no mutants yet, you'll still choose something to pass through, but you just provide a slanted playing field where if you're a group, you're going to probably get transferred. If you're a cell, you have a much lower probability. So yeah, I mean, I think yeast they've been really good. There is a question of how far they'll go in terms of how complex can get them. And I think time will tell. Like we're starting experiments now that I hope to take through 30 or 40 years of experimental evolution, because I would feel really bad if I ended my career and I didn't see how far I could push these things. Like I want to see what kind of organisms we can evolve, what kind of alien critters we can have in test tubes after a career of evolutionary biology.
That's so cool. That yeah, I mean, I feel like those are the kinds of experiments we need. Like we need doing evolution experiments on 50-year time. Yeah, that's wonderful.
Yeah, it's gonna be neat.
Okay, well, we are just about out of time. Does anybody have closing thoughts or questions before we end?
I know we're out of time, but I did have a final sort of overarching wrap-up question, which was, is that okay?
Yeah, by all means.
Cool. So I know that the sort of standard fare, super reductionistic algorithmic model of natural selection we talked about at the beginning doesn't get at major transitions, doesn't get at evolution of hierarchical complexity that we're looking at here, that we're interested in here. But it's not like you're introducing a new mechanism that falls outside of natural selection, right? It's just focus on different kinds of constraints. Is that an accurate summary, I guess?
Yeah, I don't think that there's a lot of conflict with the standard model for hierarchical processes. It's just sort of like you're shifting you're shifting what you're saying has the heritable variation for traits of effectiveness, right? Like if the standard sort of evolutionary algorithm, that's how I describe it in my class, right? Is that if you have things which reproduce and they vary, and that variation is at least somewhat heritable, and different variants have different fitness, then your population will evolve. Like that's just the algorithm. It just works that way. It could be computer viruses or biology, anything that follows those, it meets those criteria, given sufficiently large population size, will evolve. So the thing with hierarchy, right, is like, okay, with Snowflake yeast, basically, as soon as they stick together and form groups, well, now you have kind of an additional layer in which you have potentially selection playing out at the level of cells alone, and you have selection playing out at the level of groups that contain cells. So you have this sort of multi-level hierarchy, which is introduced by the fact that that things live in confined spaces. Mitochondria live within cells. That's confined. Cells live within groups. That's confined. And so so basically, it's possible to have selection playing out at different levels all at the same time, right? You have gene drive in meiosis, where variants are unfairly finding their way into the next generation, being increasing the frequency of something at meiosis, but being counteracted by selection acting at the level of the whole, right? And so it's very possible to have all different levels of selection playing out at the same time. I mean, even for mitochondria, people have shown that yeast defective mitochondria reproduce a lot faster within a cell, but create cell-level problems. So within a cell, there's selection to be a defective mitochondria. At the cell level, there's selection not to be a defective mitochondria, because you make fewer cell-level offspring. So I think what I think is really cool and really fundamental is that it's not that hard to go from being a cellular individual, like a canonical cellular individual that's got really good at being a cell because you've been a cell for 1 billion years. And now all of a sudden you start sticking to your offspring and you form these groups. And those groups they have a life cycle, which is they get for free. Nobody has to make it. You don't need any new genes. You just stop letting go of daughter cells, and physics gives you a life cycle. And all of a sudden, mutations start arising in the context of this life cycle. And because your life cycle has a genetic bottleneck, they get separated into their own groups. And now selection is seeing these different groups with different genes, and it's selecting on them differentially. And so selection can easily switch from acting on cells to whole groups. And because of this bottleneck, whole group level traits are actually pretty big. They're pretty strong because every cell in that group has this trait, and every cell in this group has a different one. And so if the trait is how elongate your cell is, that that doesn't have much of an effect on a single cell. But in the context of this growth form, now all of a sudden, every cell is longer. You have less branch-branch interactions. Your group gets larger. Selection sees that. It acts on it. It increases in frequency quickly in the population. So you can effectively shift the level of selection and thus the level of Darwinian individuality really easily because it's easy to get life cycles that essentially give you all of the properties of the Darwinian algorithm at a higher level totally for free. I hope that was clear and not confusing.
Yeah, no, that was perfect. Thank you.
Cool. Yeah. So yeah, if you want to read like 20 more pages on that one idea, we have a paper coming out in Philosophical Transactions of the Royal Society, all about life cycles and basically how life cycles can make the transition to a higher level of selection or a higher level of individuality pretty easily and pretty clearly.
Well, I will definitely check that out.
Yeah.
Well, I do have one more quick question from you, which is that you possibly send me some of the visual material that you used, because we're going to make videos about your talk. And I would love to have access to your slides and those like that Rotifer video was just like
yes. I will definitely send it to you. My only request is that the stuff in the future directions, which is mostly unpublished, let's not put. If we can keep that stuff back, that'd be great.
Okay, can do.
But anything anything else is totally fine by me.
Great. All right. Well, thank you so very much. I think we conclude. So I'm gonna check in with the class after you leave, but you can just hit a little X and then we will be done. All right.
All right. Thanks, guys. That was a lot of fun. Thanks for inviting me.
Thanks so much.
If I ever run into you guys, like at an evolution conference or something, definitely come say hi. I would love to chat with you IRL, as they say, as the kids say these days. All right. Take care, everyone.
Farewell.
Bye.